Hello everyone. Let's study structure of DNA. In the last video lecture, we have already studied that nucleic acids are of two types, DNA and RNA. Today we will study the structure of DNA. The basic unit of any nucleic acid is a nucleotide which has three parts, a pentose sugar, nitrogenous base and phosphate group. We have connected all three and formed a nucleotide. As we are going to study the structure of DNA, we have taken a deoxyribose sugar over here. So this nucleotide will be deoxyribonucleotide, right? So let's take many units of deoxyribonucleotides, join them and form the strands of DNA. So let's start with the structure. So here we have taken two nucleotides. Now these are free nucleotides. We have to join them so that we can form the strands of DNA. The bond which helps to join the adjacent nucleotides is a phosphodiester bond. Next, let's take one more nucleotide. We can join this free nucleotide to this dinucleotide which is already produced over here. So again, the bond which connects them will be phosphodiester bond. This phosphate is attached to its sugar with the help of an ester bond or phosphoester bond. There is another ester bond formed here to join the adjacent nucleotides. That is why it's called phosphodiester bond. To join three nucleotides, we have two phosphodiester bonds. So, in this way, you can join many more units and form long strands of DNA, right? So, this is our strand number 1. Now, DNA is usually double-stranded, right? Our genetic material is DNA and it is double-stranded. So, we need two strands. Let's make the other strand. Now, the other strand will be anti-parallel, means it will run in opposite direction, right? So, that is why we have drawn the nucleotide in a flipped manner. The phosphate will always be pointing outwards and the nitrogenous bases will be facing each other. So, they will be inwards. So, let's take two nucleotides and join them in a similar manner. So, we will have phosphodiester bond which will join the nucleotides. Let's take one more nucleotide and again join them with the help of phosphodiester bond. So, here we have our second strand. Both the strands of DNA will be connected to each other with the help of specific base pairing, right? So, in the last video lecture, we have studied that this particular part of the nucleotide, that is nitrogenous base, right? Nitrogenous bases are of two types, purines and pyrimidines. Adenine and guanine are purines and thymine, cytosine and uracil are pyrimidines. Uracil is found only in RNA, so it will not be present here. We will have only cytosine and thymine, these two types of pyrimidines in DNA. Now, there is a specific complementary base pairing between these nitrogenous bases. That is, adenine A will always pair with thymine T with the help of two hydrogen bonds. Similarly, cytosine will pair up with guanine with the help of three hydrogen bonds. So, in this way, we can use these nitrogenous bases, and if you have the sequence of one strand, you can easily predict the sequence of other strand. For example, let's say this third nucleotide in the first strand has thymine T, then we can easily tell that in the second strand, what we will have here is adenine A, because T pairs with A, right? Or A pairs with T. So, both will be again connected by two hydrogen bonds. So, this is the complementary base pairing rule. Adenine pairs with thymine and guanine pairs with cytosine. So, a specific purine, which is a double ring structure, pairs with specific pyrimidine, which is a single ring structure. So, we will always have a double ring structure opposite to a single ring structure. Now, in this second base pair, cytosine is a pyrimidine, so actually it is single ring structure, and guanine is a purine, so it is double ring structure, right? This will help to maintain the uniform diameter of DNA throughout the length, right? So, distance between, like from here to here, this diameter is constant. Okay, we will study about the dimensions in few minutes. Now, let's see few more points. As you can see in this double-stranded DNA, this particular end of this first strand, here the phosphate is free, which is attached to the fifth position. So this end of the strand is called 5' end. Now look at the opposite strand. Here this third position 3' OH is free. So this is called the 3' end of this strand, right? So 5' and 3'. But as both the strands of DNA are anti-parallel, so they have opposite polarity. So look at the second strand over here. Your 5' end is here, right? And 3' end will be at the top. So both the strands of DNA are having opposite polarity. Like here you can see we have drawn a very basic representative diagram of DNA showing a ladder, right? So the backbone of the ladder, these vertical pillars will be the sugar and phosphate, right? And the steps of the ladder will be nitrogenous bases connected by hydrogen bond. Also look at the polarity. When first strand is 5' to 3', then the complementary base paired one, the other strand is 3' to 5'. This polarity will be always opposite. Hence, both the strands are anti-parallel. So, this is a very simplified version of DNA, right? But this ladder is actually twisted in helical pattern. So, we will have right-handed helical structure of typical DNA. Okay, so let's go through all the important points of DNA structure. The double helical structure of DNA, the famous double helix model of DNA was proposed by James Watson and Francis Crick in the year 1953, for which they have also received Nobel Prize in the year 1962. The Watson-Crick model actually represents the secondary structure of DNA. Whatever DNA we study, the Watson-Crick model, also the DNA which is present in our cells, this is actually the B form of DNA. There are different forms of DNA. We will study about other forms in higher classes, but right now we have to remember that whatever DNA structure we are studying is actually the B form of DNA. Okay? DNA is double-stranded. Both the strands of DNA are anti-parallel and complementary to each other, right? Nucleotides in a strand are joined by phosphodiester bond, and both the strands are connected by hydrogen bonds. So, A and T pair up by 2 hydrogen bonds and GC pair up by 3 hydrogen bonds. Also, there is one more interesting point here. For any double-stranded DNA, A will pair up with T, so the amount of adenine will be always equal to amount of thymine. Similarly, G pairs with C, so for a double-stranded DNA, the amount of guanine will be equal to cytosine. This was first understood by Erwin Chargaff, and he gave the Chargaff's base equivalence rule, or simply the Chargaff's rule. He observed that for any double-stranded DNA from any source, the amount of adenine is always equal to thymine and amount of guanine is always equal to cytosine. So if we use all this information, we can say that purines, that is A plus G, this amount will always be equal to pyrimidine, that is T plus C. Purines are equal to pyrimidines for a double-stranded DNA. Now, this will not be applicable for single-stranded DNA, because in single-stranded DNA, we will not have this base pairing, right? So, for a double-stranded DNA, as A will be equal to T and G will be equal to C, we will have purines equal to pyrimidines. This is the Chargaff's rule. Okay, and based on this Chargaff's rule, Watson-Crick would understand that there will be some complementary base pairing. Next, let's see some more points. So, now DNA actually, which we originally draw as a ladder, right? We drew it as a ladder, but actually it is twisted in right-handed manner. So, we will get a helix like this. Okay. So, one complete 360-degree turn will be like this. It is 3.4 nanometer in length. One complete turn of the DNA will be 3.4 nanometer, and this covers 10 base pairs. Okay, so this length of one turn is called the pitch of DNA. So it is 3.4 nanometer. You can convert it into different units also. Like if we have to represent it in the form of angstrom, we will write it as 34 angstrom. Each turn, like from year to year, has 10 base pairs. So if length of one turn is 3.4 nanometer and it has 10 base pairs, we can easily calculate the distance between adjacent base pairs, which is 0.34 nanometer. Simply 3.4 divided by 10 will be 0.34 nanometer. So the rise per base pair is 0.34 nanometer or 3.4 angstrom. Also, let's look at the diameter. So, throughout the length of the DNA, the diameter is uniform because a double ring Purin structure is always opposite a single ring pyrimidine structure, right? So, diameter is uniform and it is 2 nanometer, that will be 20 angstrom. Next, each strand in this double stranded structure is actually twisted in a helical staircase like fashion, right? At each step of ascent, the strand turns 36 degrees. Now, this is also very easy to understand. If one complete turn, right? This one complete turn means it will be 360 degrees. And there are 10 base pairs. So, 360 divided by 10 will be the ascent, like angle between adjacent base pairs, which is 36 degrees. Backbone of this ladder will be sugar phosphate sugar chain, and the steps of the ladder are the nitrogenous bases joined by hydrogen bonds. Always the nitrogenous bases will be pointed inwards and the phosphates will be positioned outwards, right? This is the DNA structure. Watson Crick gave this famous model, and as it is helical, right, twisted, it is called DNA double helix. Also, one more important point which you can note is we may have DNA which is linear or circular. Simply put, look at this diagram. Here we have this double-stranded DNA, right? And both the ends are free. So this is linear DNA. In eukaryotic cells, right, in the nucleus, whatever chromosomes we have, whatever DNA is present, right, that DNA is linear. Whereas if we join these ends, then it will form a circular DNA like this. So plasmid DNA, the bacterial genetic material, then we have mitochondrial DNA, chloroplast DNA, these are circular DNAs. So linear DNA will have these free ends and circular DNA will not have free ends, right? So I hope you have understood the DNA structure. Now it's time for knowledge drill. So try to answer these questions, okay? And tell me in the comments. If a double-stranded DNA has 10% adenine, if it has 10% adenine, what will be the percentage of cytosine? Next, what is the pitch, that is length of one turn of double-stranded DNA molecule? Let's meet in next video. Thank you.